Great viz for this. Well, when the mud settles. I meant between Argus and Herc. So I did drop targets that are the tail and the head. If you ever come back, but we'll see if the coordinates stick. Definitely super interesting to see how off they were. But the depth was completely on point.
So we're just going to pick up video from this end. Is that the plan?
Looks like it.
Yeah, I think so.
Okay.
I just got to fly a little higher so I don't drag the tail.
Yeah, yeah.
You come down some, Paul?
Yeah.
So I was just having a chat with Mo and asking about which baleen whales have cream-colored baleen, and minke whale could be this size, and gray whales, which can also be this size. So could be one or the other.
What's floating past us right now? Is that a...
What?
Oh, I just was seeing something floating past there in the bottom left.
Oh.
I was wondering if, yeah,
The
something
vertebrate of some
gelatinous
type?
maybe?
Yeah.
But Mo's
Salamander?
making a really good point. In 2019, we had a lot of dead gray whales. And so,
Yeah.
and I remember when we found this, that's what I was thinking about, that we'd had a lot of dead gray whales that year, and that this could be one of those whales.
Yeah. A lot of them, a lot of the gray whales, at least in our area, were
Yeah.
very thin. From the stranded animals, as well as the visuals from the live ones in the lagoons.
I'm getting yanked around a lot there. I don't know.
And
a lot of that had to do with their forage areas, not having enough ice covering, which then leads to the whole ecosystem under the ice sheet not providing enough food for the amphipods that gray whales feed on. So if there's not enough ice sheet, there's not enough food for the amphipods, and if there's not enough amphipods, there's not enough food for gray whales.
Down a bit. I'm just also, I think, right above you.
You can see I'm kind of getting this action.
Yeah.
But we also have some entanglement issues in our region, particularly in the central and northern California coast with baleen whales.
Dust. Dang it.
What do they get entangled in?
Fishing gear, mostly trap gear.
It's too low over there. I don't know. They must have been pointed down more with a camera, but you get all this basket hoopla over there.
That's fine. We can take another approach, just even higher than that. Just higher than that. The software will mostly take care of it.
Ship move underway to west.
So the sanctuaries, as well as National Marine Fisheries Service and Cal Fish and Wildlife, are working very closely with commercial and sport fisheries to greatly reduce the amount of really kind of loose, free-flowing gear that might be in the water. It's the lines up to the surface buoys that seem to be a problem, particularly with humpback whales. Humpback whales, they have these really long pectoral flippers, and when they dive, a lot of them will corkscrew down, and so it's like an airplane doing corkscrews down to the bottom. And sometimes those pectoral flippers will get entangled in the vertical lines up to from the trap on the bottom up to the buoy. Sometimes whales will get that entangled around their flukes.
Maybe we need to be zoomed in a little bit, Al.
Yeah, a little bit closer.
Oops.
I was thinking about that when the ROV was entangled in fishing gear up in Olympic Coast National Marine Sanctuary, and how handy it was that we had a knife to cut ourselves loose, and that whales do not have knives. So it's a lot harder once they get entangled for them to get loose.
Yeah, there's a fair number of them that disentangle themselves, but sometimes, especially if it's wrapped into their mouths, caught in their baleen, or if they're around a pectoral flipper, those are a little bit harder. Whales don't back up very well. And we do have a great disentanglement team that works with National Marine Fisheries Service, the Straining Network that Mo works with.
100% joy gain.
And
My Z-bias is gone. We must have had a reset there. My Z-bias
and
went
the
to 0.
Mammal
My
Straining
joy
Network.
gain went to 100. Oh, yeah, we crashed. That's what's going on. Windows up. I'm like, what the?
Could that have also caused
a
that
little
camera
crab.
flicker then? The Zeus flicker?
Yeah, we
Several little
actually,
crabs.
we had a crash.
Yeah. Because I was thinking I shouldn't have been pulling on your tail. I was
Yeah,
only
I'm like,
15 meters above you.
Okay. That
All
kind
right.
of clears things up.
There we go. That's your problem right there. And the ship move is complete. So we are
All right.
in good position.
Are you good at the top side? Or do we
Incoming
need Ben?
Grenadier.
What's that?
Do we need Ben, or are you good?
We crashed and it auto starts, but it auto starts with, like, it resets all the gains.
Uh-huh.
So
So
which
what
I'm
you-
not used to driving at that. So that's why it was herky-jerky and doing all its weirdness. This will be better now.
You always have to turn it off, turn it back on again.
Yeah.
Maybe
Okay.
that was the video blip that Al saw.
Yeah,
Yeah.
I think so.
Ah, yeah.
One more time.
Yeah, the whole skeleton, you can really see just how much has been eaten away. It just looks so old and porous. And
Thought I forgot how to drive there.
There is one squat
the
lobster.
large bone that's kind of pointing down at 7, 8 o'clock. That's the lower mandible.
Squat lobster.
And it's usually a very large, thick piece of bone. And it's just almost, I wouldn't say Swiss cheese, but it's looks very porous now.
Yeah, are the squat lobsters that we're seeing a particular species that favors whale falls, or they can show up for any sort of scavenging activity? And are they actually feeding off of anything there, or do they just happen to have enjoyed the habitat?
Good questions. I don't
Yeah, that's a good
know
question.
specifically if it's a different species. I haven't heard of that. How about you, Chad or Jan? Have you heard anything about squat
I don't-
lobsters that are
Lots of
specific to whale falls?
I don't know.
So would that mean that we lost our wraps or no?
Yeah.
Okay.
Totally. I have no idea anymore.
Yeah, that's a lower left. On the lower left, on the sea floor, is the other mandible, the left mandible. the lower, our equivalent to our lower jawbone. Crispin Little says, I don't remember squat lobsters from last year.
Okay. I do remember some squat lobster, and I remember thinking about or actually collecting one. But what I don't recall is if they were this uniform white color or if they were had some red or orange on the shell. So I was going to look back at some photos from last year to try and get at Sam's question. The 3D model isn't good for looking at stuff like that, so.
And is that anemone?
No, sitting. Is that one in the kind of mid to lower left?
Yeah, there's an anemone, but I can't tell if it's on a bone or in the sand. Kind of interesting.
No, is that the chest plate sitting kind of on top of a vertebrae? Two vertebrae?
The sternum.
Yes, that's what I was wondering about. Great. So we're going to get Amity Wood, our education and outreach coordinator for Monterey Bay Sanctuary. She's going to hopefully be able to join us via the V-Link voiceover.
So, in this view, you can see one eel pout is looking out on the right side of your view, and then in the middle, you see the tail of another eel pout sticking out of a
There's the
hole
other
in
one.
the vertebrae.
Ah.
Yeah, and I think on the other side, I saw the head of that one peeking out. Nice
Yeah.
place to hide, I guess.
Yes, we did pass over. We passed over the scapula and the humerus. It's just off to the right of our screen right now. We'll get another look at it in a little bit. And I noticed that humerus bone, if that's our kind of our upper, kind of what's connected to our shoulder, the equivalent of that bone in our body. I noticed that it was still very prominent in the original model and video from last year. But you can see that the vertebrae just has a lot of holes and looking very, very porous. Now, do the oxidex feed on the cartilage first?
I think did we drift off the side?
Yeah, I think that's good coverage of the whale for the orbit.
Want another small move?
Yeah, I'm getting yanked around.
Okay. Bridge nav. meters, 270.
Good
Jan,
morning, everybody. This is Amity.
I was just going to say to your point, Jan, last year we noticed them on the bones and not necessarily on the cartilage. What I was really interested in this year is working with Greg Rouse, who's an expert at the amphoretid worms that we found last year. They appeared to be in the mud/blubber kind of matrix halo around the whale. And so we were hoping that there might be some left, and I don't know if there are or not, but there certainly aren't any direct signs of them right now. Certainly the Ossidax are here. So it looks like we'll be able to collect those for Shanna Goffretti to do some more analysis. And last year we collected 3 or so bones with Ossidax on them, and through genetic analysis we determined that, or she determined that one was a new species. So we'll be working on a manuscript for that in the near future.
There's also a bit of hump right there. That's what if I'm trying to come along, I'll run into it.
Yeah. I've marked the slope on IPAC in case I need it.
No good.
Good morning, Amity. How are you?
I'm great. This is better than Saturday morning cartoons.
A little darker than Saturday morning cartoons.
This is like Saturday morning cartoons for
I
scientists.
was going to say, yeah. Come look at this decomposing whale skeleton with us.
I mean, we're pretty
So
funny, but
I'm
I don't
looking
know if
back.
it rises to the point of cartoon.
Yeah.
Burton's version of Saturday morning cartoon.
Well, when I was a kid, I woke up early on weekends to watch cartoons, and I woke up very early to make sure I didn't miss this. So I guess in that way, it's similar.
Yeah, I don't want to get near it with all this.
5 meters left on
But
the
I
ship.
was looking back at a video from last year to see how many and what type of squat lobster we were seeing, and I did see so far in the video one that's all white in the sediment surrounding the whale, but I haven't seen any on the whale. But now that's where we're seeing a lot of them on the skeleton itself, and I wonder if they stayed away from the skeleton because it was covered a year ago in fish and octopus. And the fish that we saw in association with the whale last year were grenadier and eelpouts. And we have seen both of those here, but just in many fewer. I think we've seen 1 or 2 grenadier and 2 eelpouts so far. In fact, it looks like a grenadier is hanging out in the shadows above the skeleton of the whale in our current view.
Yeah, I remember last year too, we also had the bacterial mats that were kind of in the mud sediment around mostly kind of the head of the whale, but I don't see too much of that now.
We are curious what's the
Got another
yellow.
one?
I just want to be there.
I know, you're like at the end of the leash, just like a little more,
Yeah.
a little more.
Got another ship move?
Well, I don't know.
Yeah, so the last
Things
thing
are so slow.
visually we need to do is, just do a pass from tail to head and then head to tail over the top. And then that'll be it for the imagery, and then we can
Oh, you
start.
think we got enough?
I think we got enough for the orbit. It's just going over the top now, both directions, to kind of fill in the top blanks.
Yeah, we got to wait for the...
Oh yeah, I know.
Unfortunately, you can see how I'm kind of getting yanked up and down. Like, whipped on the
Chad,
end of the leash.
do we have lasers on an accurate measurement of the length?
I actually haven't gone through and done that. It's just the initial estimate was 5 to 6 meters, but I don't think anyone's broken it down to get a really precise measurement.
Paul, your camera slide.
So,
all the way down,
but do we
right?
have that?
Like we're getting anywhere.
Sorry, I couldn't hear you, Al.
You are still tilted all the way down in the camera slide, right?
Yeah, pretty much. No, I'm kind of
You know what I'm saying?
60 degrees or so.
Yeah, we're not...
Ship's moved quite a bit since we got on site then.
Give me 10 anyway. I mean, worst case, we go over it that way, right?
Yeah, you were tilted all the way down before, and the whale was at the bottom of frame.
Won't 270 pull me backwards this way? Am I crazy? Because I'm facing 90 right now.
I'm here. No, I'm facing south, right? Yeah. So we're looking at the from the west, looking east with Argus. So we want Argus to go to. So I think you've been making a move in the wrong direction. Yeah. So I'm facing south, right? And Argus is 090. So really, you want to go on Argus's bearing. So that's what's going on. We're getting further away. So tell them to cancel that move. We need to go the opposite way, twice that. No. So right now, we just need to go Argus's bearing, right?
Yeah.
From Argus.
But now I'm getting turned around. So I think 090 was the...
Well, opposite of Argus's bearing, because it's...
Sure.
The pointy end is... No. We want to go Argus's bearing. Sorry. The camera is looking in the direction of the compass. So, yeah, I'm getting dragged off of the site.
Okay, yeah, Argus is bearing for me right now. Should be 090. That's why I'm getting a little mixed up here. Okay, we're gonna go. What I have is that we should go north to get back on the whale fall.
Isn't it east?
Well, what was the move you just made?
I was going west, but if we go north, we'll be back on top of the whale fall. To get back up here.
I think it's just...
East.
I mean, we're pointed at the whale fall
Right.
here, and it's east.
Okay.
But
Yeah.
Okay.
And we're getting dragged off of it. So the move we made, it was bad.
It'll be interesting to take a look at the surrounding area and see if there are any other scattered bones that may have been dragged off or pieces of
it looks
alien
like
plates.
you came up a bit, Bob. Do you want me to make any changes, or
I'll
just hold?
I'll come down. Dang it.
While we're waiting, I could come around and we could zero the tether.
Yeah, why don't we do that?
Do I go? I
Do
guess the
we
question
know
is,
which way? Yeah, which way to go around? All right,
not confidently.
I'm gonna come back, look at it while we're waiting around here.
Yeah, this is Amity. Just to let folks know as well, if they're interested, we do have the chat feature available on nautiluslive.org. So if you are on the website, you want to shoot us a question or let us know where you're tuning in from, we'd love to see it. So go ahead.
So there should be half a wrap one way or another right now, right?
Yeah.
Ship move is complete.
Thank you, Amity, for that. It's always nice to hear chats from the public as well as the scientists, but I always enjoy the questions from the outside world.
Yeah, and this whale fall was very popular viewing last year, and I'm hoping that
Well, I think we needed to
people are
cancel
finding
out
it,
the
that
last
we're
one
back.
we did, right? That was that. So we had gone from here over to there, and then you came back there. Yeah. Okay. All right. You want to get straightened out, and then we'll.
Yeah, which way do we want to go? Clockwise or counterclockwise?
Don't know. Just pick one or the other. One's gonna be right, and the other's wrong. So do the right one.
If you're wondering what the ROV pilots are working on, they have to make sure that the tether that connects Hercules and Argus is always in good condition and not getting tangled up. So they are checking and making sure that it's not twisted.
Yeah,
Okay.
it can
So we're
sometimes
gonna
be.
zero reps, right?
Yep.
Zeroing 6-8. And then I'm zeroing my reps straight out. There we go. 0. Okay, come back
All right.
around.
Copy that.
Yeah. If you've ever tried to disentangle your, get the kinks out of your garden hose, think of your garden hose being about 2 miles long and trying to get one kink out of it. So it can be a challenge. And that's our lifeline to the ROV. So we have to make certain that all the fiber optics don't get kinked.
Okay.
There's
Daisy
both the
chain also
video
looks a
and
little
the
bit loose on the
What?
rear. The daisy chain looks a little bit loose on the rear cam. Maybe that's just normal when it's bending this much, but
Yeah, I don't think it's that bad.
okay.
Yeah.
Oh.
Do you not have auto heading on, or what happened there?
We're just getting yanked right now.
You what?
We were just getting yanked right there.
But were you in auto heading, or?
I was on. Do you want me to turn it off so we don't,
No.
doesn't
No.
pull on you?
Oh boy.
Ship move complete.
Okay.
Also, Bob, you've got a notice here. Topside restarted 1653, but we're not even there yet. 1653 hasn't happened yet.
Oh, that's interesting.
It's the future.
Well, if topside restarts at 1653, we'll need to figure that out.
I hope not.
I'm not sure if I like.
I think one is enough.
Yeah, I'm not sure if I like the computer's foretelling.
Sam, type in Powerball number.
Yeah, 1653.
Question mark. Hey, wait, yeah.
Okay, so I should be up in here because
Yeah.
that's where all my trails are, right?
Correct. And when we get you settled, I might try a DVL reset again because this is not tracking.
All right, we've got lots of questions coming in here. Keep them coming. People tuning in from Las
Right, but I'm
Cruces, New
still at the end of my
Mexico.
leash, so
Another step. Like a northeast step.
yeah.
Roger. Bridge nav. Uh, 10 meters, 045.
So Chad, it sounds like when we get back to the whale, we're going to take some video from above, pointed down, going head to tail, tail to head kind of
Yeah,
thing. And then
it'll
we're going to
fill in
want
a
to do
lot
some
of
of those.
those blanks for the 3D model.
Great. And then we'll want to do some of the collections we've been planning, right?
Yes.
All right. And here it is. So Amity, you were saying where a few folks were tuning in from.
Yeah, across the world actually, Germany and Norway, across the country from Florida, Baltimore, New York. Got lots
That's
of questions
great. Well,
about the decomposition of the whale, and what are some of the factors that we know that determine how fast these whales do decompose? I would imagine it has to do with the level of scavengers as well. Maybe temperature.
Are we still moving the ship?
Yeah, we have 3 meters to go.
Chad, do you want to take that one, or you want me
Yeah,
to?
I mean, there's so many factors, like you mentioned there, Amity, not just the scavengers, but how much decomposition is going on in terms of bacterial, the bone-eating worms, all the other things. What's also important is temperature and, of course, the depth that it's at, which will kind of say what the temperature is. There's also the possibility of whale falls falling within the oxygen minimum zone, which things are going to be different. There's going to be a different array of scavengers at every depth level. So I think every whale fall is unique in those variables. But
Half meter.
this whale fall, which we discovered almost exactly a year ago, I think we're 6
want
days
another
shy of the 1-year anniversary. The rib bones were still visible. There was still heart tissue in the aorta. The baleen was still there. It looked like a full articulated skeleton with still a decent amount of flesh on it. And it was estimated to be about 4 months old from the experts, and I am no expert on this, but these reefs can last, these bone reefs can last for many, many, many years. There's only been about 75 whale falls documented in the world, but we know there's many, many more of them, in the hundreds of thousands most likely worldwide. But this is a really rare opportunity. Not only was it rare to discover a whale fall that was relatively fresh, but it's rare to come back to a natural whale fall like we are today. So we can look at all this imagery and some of the collections we're going to make today, and then make some real observations about the difference in the decomposition and scavenging of this whale over a 12-month period of time.
I would imagine that rate does change as a lot of the fleshy material gets scavenged on as
Al, you
well.
want to zoom in while we're waiting?
Yes. I probably want to go in. I'm trying to fix an audio problem too at the same time, so keep me apprised.
We don't want to go on too much because we're kind of getting jerked around.
Oh, great, now we can start to see that baleen. So last year it was creamy. It was still all attached to the jaw. Or now you can see it's been scattered around, and it's turning a darker color, and we mostly are just seeing the darker plates, and
That
we're not
little,
seeing all the fibers. The free fibers at the end aren't
see
really
the
as
scotoplanes
nearly
there,
as many.
Jen?
What's that?
There's little scotoplanes there, little sea pig that just came.
Yeah,
Oh,
little
yeah.
sea pigs coming in, yeah.
Oh, and some actually quite small squat lobster in and amongst the baleen, as well as some of the larger ones.
What's the pink? What's the pink thing?
I think that's a cucumber. On the right side, Jan? Looks like a
Ship
cucumber.
move just
Yeah,
about
lower
complete.
right.
You got complete?
Yep. Half meter.
Oh, are there some worms in there, maybe? And then amongst the baleen?
Starting to look better, but
Might be some
Looks like
tubes sticking out of the
anemones.
sediments.
How about then up a little bit?
Oh,
I see some tubes sticking out of the
Actually, can I
sediments.
reset the
It's whether or not
DVL?
there's still worms inside.
you know where it is.
Well, this cluster is definitely more promising than up here.
Is it?
Well, we're not getting any.
Yeah, okay.
That also
looks
makes
like
more
a
sense
So is
for
it
how
cluster
you're
of
getting pulled.
worms. Looks like a cluster of worms off. It's off camera now, but it's lower right.
Yeah.
I think we shouldn't have gone quite to 4 or 5.
So maybe back a little bit.
What? More. I think you need
Yeah.
to go like down here.
Do-
1, 3, 5.
Yeah, definitely. The baleen has
down.
been scattered a
125.
decent distance from the skeleton.
Maybe even more than that. Maybe like 10
Yeah, I was
meters,
noticing
1, 5
when
is
we were moving away to do some umbilical maintenance, they do seem to go pretty far off in this direction. About a 2 o'clock direction from here.
Jan, do you know how many plates, individual plates, there are in the mouth of a baleen whale?
You zoom back out, Al?
Approximately?
Depends on the species, and I bet you Mo actually knows exactly for
So many pushes
minke
Zoom in
past
past
versus
the junk.
gray. I'll check the science chat. Or maybe Kirsten. Oh, she doesn't know exactly, so we can look it up.
I mean, it seems like quite a few.
Yeah, it is quite a
field
few.
of baleen.
Hold on. I am going to step away and take a look at my dead marine mammal book. Hold on.
I mean, everyone has a dead marine mammal book, right?
I've got 2.
That's great that she has one. So right now we're taking some videos starting at the head of the whale and running down the middle of it all the way to the tail. And this video is going to contribute to scientific study of the whale fall, as well as the creation of a three-dimensional model that we can compare to last year's three-dimensional model and look at the changes. We do see an eel
Thank you,
pout
Mo.
hiding under one of the ribs on the right side. That's a fish. And And we have a squat lobster party going on the head. Thank you for noticing that, Kirsten. Kirsten, I mean. Last year, in looking back over video, the squat lobsters were not on the skeleton, at least not that I could see, but more around the edges and the sediments, and now they've been able to move on to the actual bones, probably with the fact that none of the larger fish and octopus are really here anymore.
So Mo's looked it up. 130 to 180 baleen plates on each side of the upper jaw for gray whales. And then minke whales have more, 200 to 300 baleen plates on either side. So we could actually take a look at the model, and if there's 130-ish, could be a gray whale, and if there's over 200, it'll be a minke whale. So only we can tell what species it is. They don't seem to have an overlap in number of plates, so we could probably figure that out from the original model.
Ship move complete.
Hey, Jan, I know that there are, basically the baleen is made of keratin, like our fingernails, but aren't different species don't they have different types of almost like protein within the baleen? Because I know there's variations of different colors of the baleen.
Excuse me, Amity, could you move your mic a little bit away from your mouth?
How's that sound?
Ooh, a little bit too much. Split the difference, please. I know, a funny way to set levels, isn't it?
Better?
That's perfect. Thank you.
Great.
Okay, yes. There's different lengths. There's different kind of the hairs. I'm using air quotes here. Hairs on the inside of the baleen. It's kind of like a brush or very dense comb where the baleen splits off and kind of is more frilly. And so larger whales like right whales, which don't really have throat, they have really long baleen and they open up their mouth really wide, and the kind of frilliness of the baleen on the inside is very, very fine, whereas in gray whales, it's kind of tough, coarser hair. And again, air quotes with the hair. And they come in different colors too. Fin whales will have, adult fin whales will have white or cream color on one side and dark, kind of almost an olive dark, very, very dark olive green on the other side. Whereas all the other whales, they have pretty uniform color in their baleen. And as far as, what was your other question about the keratin content?
No, I was just reviewing the protein that makes up the baleen. So it's not bone, but it's keratin.
Yes, yes, it's very similar to our fingernails, that type of protein.
So now we see this dark coloration on the baleen that we didn't see last year. Is it because the baleen was still in intact rows within the jaw that we couldn't see the dark coloration, or do you think it's actually changed color as it's being consumed and degraded? Any thoughts on that?
Oh, it changed color, yeah.
Okay. So Kirsten was thinking that counting the baleen from the video might be something that some of our viewers would find to be a fun challenge. not the current video, but from last year's video when it was still in associated with the jawbones. Last night I heard there was also discussion of the fact that all the YouTube videos are available on Nautilus Live YouTube, and that people were showing interest in the chat about helping to count octopus. So there's a lot of things that interested viewers could do with this video to contribute to their scientific understanding of what's going on here.
All right. What's our what's our predicted ascent duration today?
Looking for a time?
Is it 3 hours or so?
From this depth, yeah, a little bit over 3
A
hours.
little bit over 3 hours?
Yeah.
Okay, and I know that we wanted to be on deck no later than 1600. So
Cool.
we should be leaving the floor absolutely no later than like maybe 12:30 or something like that.
Yep. Yeah, it's about an hour every 1,000 meters.
Yeah, we're a little bit more. So 12:30 would be
your speed on.
good. And
You should be
it is
basing
now
on like 13,
almost 10 o'clock.
13 meters per minute.
Okay, I'll calculate
18
that.
was way too optimistic. We did
And
between
maybe
13 and 15
Chad
on the
and
last dive.
Sam, maybe an extra half hour to get all the vehicles on deck?
Yeah, so
Or is
what,
that within that 3 hours?
what I was
mean,
hoping
it's what we're
earlier when
doing,
we were doing
you know?
the
It's whatever the configuration of the vehicle is, and it ends up, yeah.
Roger, let me calculate it.
when we were doing the octo cone, it took a little longer than I was hoping. Still was a great dive, don't get me wrong. I was just trying to build another 4 hours into this dive, but we're clearly not going to get those 4 hours because we haven't even started collecting samples yet. But if we could play a little on the conservative side and maybe try to leave by, you know, noon at the latest, but earlier if we can. But I'm hoping an hour and a half or 2 hours gives us enough time to sample what we want here. Just keeping an eye on the clock. And so I'm going to just kind of watch it and zone out because I'm a little tired. And this is Jen's shift, but she knows what we're trying to collect here. Ossidax, amphoretid worms. We had a request for baleen plate, unarticulated smaller bones, the caudal vertebrae potentially. So we got a laundry list here. And then, of course, a lot of nice shots, right? Land and zoom, land and zoom, land and zoom.
Yeah, and we're going to want the lasers on for some of those so we can get some size measurements as well, I assume, of some of these bones that we're not collecting. But it seems to me we've completed our video needs for the 3D. Okay, so we can start looking for these, I think we need to look for worms in the sediments as well as bones that are the right size for collection, that hopefully some of which have the ossidax on them. And Jan, you were noting a few bones along the way here that are of particular interest. So maybe we could start there. We talked about caudal vertebrae. Have you seen any here that you would like to take a closer look?
Yeah, and don't forget the auditory bullae that are very well exposed. They might be difficult to get off, but those are pretty nice for a museum collection, and I don't see very many worms on that. So a vertebrae for the museum, auditory bullae for the museum, and I think maybe attempting that last, because it could be the most difficult thing to get off of the skeleton. A few pieces of baleen for the museum also. And then for the worms, some smaller bones for those also, because I don't think I think we have to be careful about the size, because the compartments in the bio boxes are kind of small.
So Bob,
So I think
thought
I think
might
the
have
belay will
seen-
fit in. Yeah.
Sorry, go ahead. I was just going to say, while
Go
we're here
ahead.
at the head end of the whale, I thought I might have seen some tube worms in this muddy patch below the lower mandible. And so maybe if we could zoom in here, we'd be able to get some baleen and potentially slurp some of those worms if they are present here. And see whether or not the mandible, it looks like it might have some of the ossidax on it, and it might be frail enough that we could break some off.
Yeah, that would be good. Just
Bob?
need to measure it and ask Steve about the size of the bio boxes that are available. I guess
Yes.
we have some coral samples from earlier in the dive.
Yeah, so we don't have any boxes full right now. We only have 3 slurp jars full, so the boxes are wide open. In terms of size, the starboard box, the outer compartments are going to be your largest bet, but the forward boxes are also quite large. So if you have small pieces of bone fragment, those might go in the starboard box, but in the interior compartments.
So, Bob, can we head towards that patch of mud that's to the left side of the screen, below the mandible, and that kind of has a ring of baleen around it?
Watch lead, you might need to repeat that. There was just some internal conversation here.
Yeah, sorry, Jen. We were trying to figure out some resets that were happening. Bob, are you still on SPL?
Okay.
I am now.
Okay, I think our first location that we'd like to look more closely and hopefully make some collections are to the very left, bottom left of the screen. There's you can see the jawbone. Right below that is kind of an open patch of mud. And then there's some baleen ringing around that. And earlier I thought I might have seen some of the tube worms in that mud. So I'd like to try to zoom in there and take a closer look and try and identify some baleen, potentially worms in the mud, and maybe a piece of jawbone with ossidax that might be either loose or easy to break off.
Yeah, it looks like the tip of that mandible, just in the lower left, has a lot of ossidax on it. There might be smaller vertebrae at the caudal end. Oh, look at those nice little disarticulated pieces of mandible with some ossidax. Very good.
Yeah.
That should be swollen.
Okay, good.
Do we need lasers on?
Yeah, we might as well turn them on at this point, I think. And then Steve will need you to help us with biobox size as we're looking at things and make sure we stay in a realistic size class for what we're looking at.
Not a problem. I've also instructed video to put Telestrator on SAT3, so I'm going to draw on things as I think you're talking about them, see if those are the things you definitely want to collect. So just keep an eye on SAT3 as we do these collections.
Okay, thank you. That'll be super helpful. And then as far as slurping worms, if we find worms, how many slurps do we have open?
We have four buckets open.
How far you want to go,
4
Bob?
buckets open. Okay.
Past the light.
Oh yeah, that's brighter. Thank you.
For the scientists, I'm bouncing back and forth on one screen between a close-up of HerCam and the science chat. So I will try and monitor science chat.
Well, it seems to me these pieces of jawbone at the very tip of the jaw, kind of going almost out of view, the bottom, center, of my screen, which I know is a little lag behind the ship, appear to have a lot of ossidax and squat lobster on them.
Yeah, and it looks like that
from the lasers, maybe 10 centimeter chunks.
10 centimeters, yeah.
So, and then does it matter the size of the baleen? Mo, do you want some of these smaller pieces? Okay. Like, Steve, could you circle? Well, I guess Mo won't be able to see that. But I'm talking about the piece of baleen that is, if you follow the jawbone, it would be directly in line with the jawbone.
Yep,
It's on
I
the
see what you're
smaller
talking
side.
about, kind of in this area.
Jan, do you think that that piece of baleen would be good?
Let me take
Yeah, I'm talking about that area.
Smaller pieces are okay,
Bob,
but
would
the
you do that
longest ones
breakup
will
again?
give more time series.
We just got that breakup at Hercules again, so we'll check your top side.
58. What was our prediction? Was 53.
No, it's different.
So Jan, there's a much longer one to the right of the smaller piece.
A larger loss of signal.
That's probably
to the right. Yeah, to the right of that.
25
that
centimeters. Is that too big, Steve?
No, that'll fit in just about any box. That might even be able to go vertically into the starboard inner boxes, but yeah.
Yes, the one you put the arrow on is the one I was looking at.
Yeah, that looks kind of twisted, this one, yeah.
Sam, what was that time before that hadn't come yet?
Yeah, that's a good one, Steve.
Okay, so we see a couple things here. And Mo, can you confirm that these bones on the jaw are okay? And if so, then we can set down here. And at the top of the screen, definitely are some tubes there as far as worms. I can't tell right. I don't see any open per se, but okay, so bones on the mandible would work too. So
yeah.
You want to zoom in close there, Al?
There we go. So here we have a nice view of a piece of bone with bone-eating worms waving in the current and a bunch of small squat lobster. So let's grab that piece of bone, and just for anyone who's watching who may be interested to know about, we have permission and permits and everything that allow for us to collect some of these bones, because if you were to encounter mammal bones on the beach or marine mammal bones on the beach, you're not supposed to take them home or keep them. But this use for science has been all cleared. I know that last year there were some people watching who knew about that, and I just wanted to make that clear as well.
Yeah, that's a good point, Jen.
So let's go ahead and, let's grab this piece of bone.
So would you be interested
put it in one of the...
in sucking on it first to get the inhabitants before we pick it up? Because they're all going to bail, right? I don't know if you want those guys.
As far as I know, we don't have anyone interested in the squat lobster, and we want the worms to stay in good condition, so I don't think we should disturb it with the slurp.
And this is going starboard side?
Yeah, can we zoom out just to get a laser shot once more? Just like yeah, this I think that'll fit in one of the small compartments of the starboard box. It's less than 10 centimeters in diameter, about.
And then once we pick it up, it'd be great to rotate it as well, so we get a nice view of it before we put it in the bio box. Thank you.